Thank you, Lee. It's really a great honor to be here and with this wonderful panel and to be part of this. It's exciting. It's challenging, I must say, but it's worth it. The other thing that makes it so a good occasion for me is that this is the Writer Center, and it's filled with writers. But writing, as you know, covers everything. It doesn't just cover novels. It doesn't just cover essays. It covers just about every form of expression. Even thinking is a form of writing in your mind. And to be able to see science now represented in the Writer Center, and this is the second of such talks, of such panels, I find it's rewarding and it's important, I think. So it's a great pleasure. Now, science is, it's got a sort of a reputation, I guess, for being factual. It's just a bunch of facts. It's a person say, what's a scientist? Well, a scientist is a person who explains the laws of nature, facts. If a person is having an argument with somebody, they'll say, oh, what are you talking about? This isn't fiction, this is science, when they mean it's an absolute truth. This is fact. Well, in fact, it's a little more complex than that. And ever since I closed my laboratory and have devoted my time to writing and wrote short stories, novels, etc., I find that the processes of science and the processes of writing are just incredibly similar in many ways. People would ask me, how can you spend the day in this laboratory and come home and write some crazy short story about somebody who doesn't exist? Well, it turns out that the problems I faced in the laboratory were not necessarily so different than the problems I faced in writing a piece of fiction. Science is not just a barrel of facts. Science is, the facts by themselves is not science, I would say. The facts are the raw material to weave into stories that are appropriate for the time and that are understandable. You have a fact here, you have a fact there, you have a fact there. Those are the observations, maybe the pictures you took when you were on a vacation. But what's in between these? Nothing. You have to link them. Otherwise, if you threw them all in a barrel and said, here's 4 facts, I mean, where are you? Fine, but they don't make sense. So you have no choice but to use your imagination to have a link. Now you try to make that true, but why is it that every time a new observation comes up, somebody says, oh my God, I've got to change my story a little bit. It's not quite like I thought. And another piece of information, if you look at any work of science and you trace it for 200 years, you'll find that it's gone through 15 different stories. How could that be if every time they presented the facts it was finished? It's not. It's the story. And so what I'm going to do today, if I can figure out this high tech, is the one point I would like to make that I think is kind of important as a background is that science is not nature. Science is an expression of selected observations, like I've said. The facts, just as art and writing are expressions of what we see and what we hear and what we feel. It's simply a story in that sense. If it were nature is nature. The facts are simply an expression, a human expression, an expression to the best of our ability. Now, what I thought I would do today is take a chance. This is a science panel. And so I'm going to take you through a short journey of science. And as an example, I want to use my own research because that's what I know the best. And I want to show you how we started, where we were going, and how this became a story that you never would have imagined that it came. So bear with me, and I'm just going to take you through the absolute reality of what I was doing in the lab. So it starts off with a mouse eye, in the sense that this is just a lens. It's what you have in all your eyes, except I don't. I have cataract extraction of the summer. But in any case, you have the lenses, and you have the cornea, and the light goes through, and it is a major structure. It's a very interesting one to study because it's completely encapsulated. It's completely cellular. There's really no fluid in here. In fact, there's less fluid in a lens than in any tissue of the body. But despite the fact that it's packed with cells, like your fingers, your hands, your feet, here it is when you take it out of the eye, look, it's transparent. I mean, that's already a remarkable adaptation. This isn't just any old tissue. If you cut your finger off and you put it on the Petri dish, which I don't recommend, it would in fact not be transparent. When I first took my first lens out, I put it in a Petri dish and I couldn't find it because I figured, where the heck, I know I put it there. And then I saw a little magnification under the thing and I looked at that. There was my lens, little magnifying transparency. So it's transparent. If you get a cataract, it's opaque. You got to take it out. Fortunately, it's not a medical problem today because you can put in artificial lenses perfectly well. Now there's another specialization of the lens, which is very important. That is that when the light goes through it, here is a laser beam that's going through a fish lens. And all lenses of all species are transparent and they look basically the same. The light goes through and it's bent in a very, very precise way so that it forms an absolutely perfect focus, and that's why you can see me in focus today. It just bends properly, and the photoreceptor cells, those which are visiting, they're right there. Incredible adaptation of nature. And if you put in a quartz bead, which would look the same to the naked eye, you can see that there's all kind of scattering, and it doesn't form quite an even thing, and it would be a big mess. You'd have a little firecracker in your eye. Bad news. So you have a lens. They're the same in all species. They're transparent, and they bend or refract light in a perfect way. Now, there's a conservation rule that all scientists go, and I think I should have called it a dogma, because it's true in all cases, and it's what made me start studying the lens. One of the reasons is that there's, in nature, the evolution, you'd have a highly specialized task, you develop a highly specialized protein to do it. An example that you all know is hemoglobin and oxygen transport. We know that if you have hemoglobin, it's transporting the oxygen. That's its highly specialized function. Antibodies and immunity. We all know that. We get sick, we make antibodies. These are proteins. They fight the immunity. Great success. So what did I expect when I started this work? I expected that crystallins, which are what you call the proteins in a lens, but the only reason that it's a crystal clear lens, the crystallins or lens proteins, I would expect to see are specialized, and I would learn about vision. Simple. I have a simple system that's transparent. It's got two facts, and it's full of these proteins. I can study them, and I can learn about how the lens works. But here I'm going. I'm writing this story. It's all as expected, but not at all. We started looking at turtles and different animals, humans, mice, and fish. They were filled with a protein. I'll call it A for simplicity. But then we looked at a guinea pig. It had a completely different protein, nothing connected to A. I mean, it would be totally different. A chicken, totally different protein, no relationship. It's like having steak for dinner or spaghetti. A duck, like the chicken, should be similar. Don't count on it. Totally different protein. If you look at the hemoglobin in these animals, it would all be basically the same. They'd be changing a little bit to adapt to the conditions. But no, here we have a broken rule. So suddenly, my story was going astray. And to give you an example of how each of these proteins can do it, you saw the beautiful bending of the fish, of the light, the refraction, which I repeat here. And here's a squid lens, where the squid of the fish, not a fish, cephalopod, where it is transparent, the light goes through, it focuses, right? But that protein has nothing to do with the proteins in the fish. This is what's called gamma crystalline, this is S crystalline. They're like Venus and Mars, and yet they have exactly the same function. So if I just threw that in a barrel and said, this is science, what would you be? You know, you'd want to go to a good movie instead. Now it got even more crazy. There's a monkey wrench thrown into the system in that these proteins, these lens crystallins, also perform entirely different functions when they appear in different places, such as metabolism or stress protection. So you have 80% of the protein bending light and being transparent in the lens, and you put it in the liver and it's having some kind of enzymatic function. In fact, if you mutate a particular protein, that makes the lens not work well. In certain cases is one where you get mental retardation in children. Same protein, exactly. What kind of story? Now, say I put that in my barrel of facts. I mean, where would I be? Is that science? It's nuts. So they had major plot changes. That's what I had. I had a plot, study a specialized tissue, specialized protein, learn how it works. But no, My plot went bananas. I had different proteins performing exactly the same function in lenses. And I had exactly the same protein perform multiple functions in other places. It was just not consistent. And the only way to put this together is to sort of write a story. I mean, it's really not that simple. Let me tell you sort of as an analogy what this means. It means that you could have a lawyer, no pun intended here, sorry. A lawyer or a power mechanic would be the same person. Or a physician and a tennis teacher would be the same person. Or a house and a wristwatch would be the same object. It's really crazy. But now let's start writing a story about this. This is an incredible paradox. And paradoxes are interesting. The paradox is that you have specialization, transparency and bending light, but you also have diversity, the different proteins, and it does different things. And normally you'd say, when you specialize, you don't get diverse. Come on, broaden up, take liberal arts education. But no, specialization in protein and diversity of doing many different things are going hand in hand. And I called it, in an article, I think, that the lens is a specialist's nightmare. So already the story gets very interesting. It's not just the facts. And so I wrote a story about this, basically, and I called it gene sharing. And gene sharing, I meant, is that the same gene can specialize for more than one function. It does, the gene makes the protein, it does multiple different things. So I thought it was gene sharing. So what did I do in terms of writing a novel or writing a science book? The first chapter that I started is how have lens proteins specialized for vision? That's what I wanted to study, simple enough. But the last chapter of the book turns out, how do genes specialize simultaneously for more than one function during evolution? It had nothing to do with vision. In fact, it took me into the whole field of evolution. It broadened everything. Nothing was the same. I started with vision, but I ended with evolution and gene expression. That, to me, is not just facts, and it shows the process of what a scientist goes through, how he's got to be writing stories, how he's got to be constantly malleable, how the stories end. And so now comes the next question. And the next question is, and everybody asked me at every seminar I gave, oh yeah, well, this lens, small little thing, it's just, it's an aberrant thing. It's not the way you really have in all the other proteins, not hemoglobin, not antibodies. So it's a good point. So I read about 2,000 papers, or maybe more, to try to analyze this. And what I found is that almost every protein that I looked at had multiple functions, very different functions. Hemoglobin has enzymatic functions, all kinds of things. They're all over the place. Nature is pragmatic. It's like if you have the door flying open and you can't close it, you might pick up a chair and hold it. Well, you're sitting in a chair, it's holding a door, or you want to change a light bulb, you'll stand in the chair and change a light bulb. It's already doing 3 things. Yeah. If it's not doing 4 things by putting 2 chairs and a plank across it when you clean the restroom. It's doing all these things because there's the protein, it can do it, it doesn't. It's pragmatic. So the story now has gone from specialization to pragmatism. And I wrote a book on it, there's the book. Now, I'll end up by just saying that I learned throughout this work that jellyfish have eyes. And that floored me. I mean, I just like stuff like that. Jellyfish have eyes? I asked the director of the NEI about eyes and jellyfish, and he was ready to fire me at the moment. He thought I was absolutely crazy. But I found that it was studyable, so I went to the swamps of La Parguera, and I show this because it's such a beautiful slide, and there I was in that boat trying to hunt down jellyfish and to study their eyes. And what they have is amazing, and I'm almost finished here, but here's the jellyfish. But here are these little black spots. And these little black spots have these enormously interesting structures where they have 6 eyes, different types, and you see this clear bulb here? That's your clear cellular lens. It's exactly like yours or mine, but again, it uses an entirely different protein. So now let's take the storytelling to the extreme as I end. I said, this is worth a book. So I wrote a novel, Jellyfish Have Eyes. And I don't say they'll be here selling it, but Jellyfish Have Eyes takes you to the next and last step of what I was talking about, science. That's fiction and that's storytelling. because it's about speculation. And speculation is not fact. Speculation is imagination. Speculation is a novel. Speculation is everything that gets our motors going. Is it that way? And so my protagonist speculated that jellyfish have, I mean, the science in it is correct, but it's in the future, and jellyfish interact, and jellyfish have memory, and jellyfish do all kinds of things. But that's speculation. Yeah, but wait, it sounds crazy. But what's the best work on memory these days? It was by Eric Kandela won the Nobel Prize for studying memory in snails. Do snails have memory? Do they think? There's a question. Does a snail think? Well, you can't rule it out because the most we know about memory is on snails. Well, my protagonist wonders about jellyfish too. So such a question as how did thinking evolve is not a factual thing. It's a novel, but yet it goes down and you've got to provide the story and you look for the truth in it and you develop it and you do the best you can. And so science is a story just like the novel is a story. And it's really great fun. Thank you.